Hello everybody and welcome to the second part of the application of pollen analysis lecture. Today we are going to talk about the ways how to reconstruct past vegetation, then few things how human impact can be detected from pollen records. Then I'm going to talk about a little bit about how to reconstruct actual land cover, as the pollen percentage sometimes might be over or underrepresenting some taxa. So there's been created different models to reconstruct the actual land cover, and I'm going to present one of the models. Then I'm going to touch few topics on the current and future trends in paleoecological research where pollen analysis is used. And then at the end of the lecture, I'm going to give you a short introduction to non-pollen palynomorphs and how to use those to reconstruct past environments. So reconstructing past vegetation, it's largely based on the knowledge of modern analogs. So basically the idea that the modern pollen spectra from modern plant communities can be compared to fossil pollen assemblages. So basically, when we know what kind of vegetation is growing at the moment in certain area and what the pollen record from that area looks like, then we can go and take a fossil pollen diagram, and based on these modern analogs, we can make interpretation how the environment have looked and how the vegetation have looked based on the fossil pollen. So basically, the idea is that the fossil pollen reflects similar vegetation than at the moment in modern situation. And some examples, for example, if we have in pollen diagram, there's tree species such as spruce, pine, birch, or alder, which are usually common species in boreal forest. So we can deduct that then the forest growing at that time is most probably being boreal forest. Similar, if there's a pollen record showing lime, oak, beech, or other temperate broadleaf forest trees, then we can make the deduction that the forest that has been growing has been temperate broadleaf forests. Or if in the pollen diagram there is some alpine species or species that are usually growing in Arctic environments, we can make the interpretation that probably then the vegetation has been more like tundra-like or more typical to higher alpine altitudes. So this is the basic idea behind the reconstructing the vegetation. Here is one example how that has compiled large data sets of modern pollen samples and modern pollen assemblages, and basically used the modern pollen data to create this kind of calibration set that can be used, for example, when making climate reconstructions, and you will probably hear more about reconstructing climate in Jon's lecture later on the course. But basically what you can see here is a big set, large set of samples and sites where the pollen records have been taken and analyzed. And here you can see the vegetation zones on the map. And here the pollen assemblages have been put in order into the diagram so that like the samples from one area, you can see them here, for example, showing the mixed forest, or then the assemblages that have the pollen which is typical to taiga forest, it's in this zone and so on. And then the northernmost sites you can see in the pollen diagram showing the pollen that is typical in tundra vegetation. So this is showing quite well how the pollen records actually are comparable and reflect quite well at the modern vegetation. Here is one example of vegetation reconstruction based on fossil pollen record. And from this diagram, we can quite clearly see the changes that has occurred in the forest dynamics and in the vegetation during the late glacial, early Holocene transition. And here we can see quite notable change in the vegetation during the younger Trias period. And we can see that there's been clear decrease in tree pollen, and then there is increase in Artemisia, and also some increase in spruce actually. But then, because this is probably because of the cooler climate conditions, which has been favored spruce, but also then is seen more like this Arctic vegetation. And this period has actually got its name from this mountain events called Trias octopetala, because the pollen and macrofossils of this species have been found from these layers, and therefore it's given the name for this period. But this is good example how the past vegetation change have been reconstructed through pollen data. Here is one other example. Here is a reconstruction of broadleaf forest cover and how it has changed throughout the Holocene. And in this study, they've divided the pollen data into land cover classes and make this kind of a reconstruction how the forest cover have changed. And from here, from the oldest time slot, around 7,000 years ago, to the youngest time slot, we can see how the amount or the abundance of broadleaf forest have been decreasing during the late Holocene and showing less broadleaf forest on the map here, which we can also see in the pollen records here. And we can see here the proportion of broadleaf forest decreasing at the same time that there's more indicators of open land. So, addition to just reconstructing the vegetation and how it's been distributed and how the vegetation communities has changed, we can also use pollen data to reconstruct the population change, and especially the change in the size of the population, if it has increased or decreased. And as from the previous lecture, you hopefully remember that this is then done using the absolute pollen analysis. So basically looking at the concentrations or influx values of the pollen record. And here is one example, which already the same, so some of the same sites that I showed you in the previous lecture, but basically an example how in northern Finland there is 2 lakes and how the vegetation has changed from early Holocene, these birch forest, to more, you can see here, increase in the slow increase in pine, but here at the first slot, it's mostly just birch forest, and then increase in the pine and decrease in the birch, more pine dominated forest, and then actually decrease in both in pine and in birch towards more this kind of Arctic forest ecotone. So we can use the pollen values to reconstruct how the population have changed. Another example we can see by comparing the pollen record from Tilia and Paicia, so basically lime and spruce, and these sites here from right to the left is going from east to west basically. So Lake Kirkkolampi is the easternmost site, and Lake Glotjärnen is the most westernmost site. And from this we can see the spread of spruce to Finland and Fennoscandia, and at the same time, when there is clear increase in the abundance of spruce and the increase in the spruce population, at the same time there is clear decrease in the lime population, and the amount of tilia pollen, it decreases clearly, and there's a change from more lime trees to spruce dominance. And one of the reasons for this, this occurs around the time that the climate started cooling around Finland, and spruce is a very good competitor. So it's taken over the habitats that tilia has been habiting before the invasion of spruce. So this is again one example how to use the pollen accumulation rates. Then there's one more example of this reconstructing the change in the population. Now this example shows the area of the occurrence of different species, and now it's represented in a diagram, basically showing the change in the area of different tree taxa. And if we look at this diagram, first here in the late glacial early Holocene, we see that the area of different tree species is quite small compared then to the later, when after the last glaciation the temperature started to rise, and also the distribution of different tree species started to expand, and we can see quite rapid increase in the area of especially temperate broadleaf trees. With spruce and fir, Eusopacia and ABS, it's been more gradual increase of the area, and also with the beech and then hornbeam, there's been more increase towards the late Holocene. And most probably the spread, the gradual spread of spruce and fir might be because of climatic change, also, or changing climate towards during the Holocene. Also beech in Fagus, it have been the climate, but then also it's been related quite strongly to that human activity might have played role in this extension of beech. But what is also interesting to notice is that around 4,000 years ago, there started decrease in the temperate trees, which is also related to human activity and deforestation in Europe. As you remember in the map showing the broadleaf forest cover in Europe, you saw that towards the modern times the abundance of the broadleaf trees were decreasing. Then we can also use pollen data to reconstruct species range shifts. As I already mentioned on the previous slide, after the last glaciation and during the Holocene, there's been big change in the tree species and species range in general, because during the last glaciation, obviously northern parts of Europe were covered with ice sheet, and trees were persisting in more southern populations. And there's been many studies of the migration rates and the migration patterns after the melting ice sheet, and also how the trees expanded back to the areas that were exposed from the ice sheet. And from pollen records, when doing research from multiple sites all over, for example, transect from east to west like spruce, or just from south to north, it is possible to detect the change of the rate of the range of some population, and also expansion or establishment of some species. And I've chosen here some examples. Here, for example, pollen data has been used to study the expansion of beech through Europe, so Fagus sylvatica. And with the different rasters, we can see the area that is estimated when beech occurred after the glaciation. So we can see that in the southern parts, it's been almost, well, all Holocene, and then gradually when we go towards north, the beech expanses. And from the pollen diagrams here, we can see that it's been first here in the sites in France, in West Germany, here in Romania, Bulgaria, and then the latest here north in northeastern Germany, and so on. Another example, again, just showing the spread of Norway spruce, so Picea apies, to Finland. So basically, about 7,000 years ago, started from the east and spread across Finland during the next 4,000 years, and then reached Sweden around 3,000 years ago. One more example of the species range shifts is the change in the occurrence of hazel. So basically, Corylus avellana, which we all know, we can buy hazelnuts from the store and eat them. And also during the Holocene, hazel trees were growing much more north or in northern locations than they do now. And also in Finland, they were, the extent of the hazel were much northern than now. And there's been many fossil findings to central Sweden, to central Finland, and also pollen records showing the spread of hazel trees during the Holocene thermal maximum, which was from 8 to 5,000 years ago. At the moment, the population of Corylus, like if in the mid-Holocene and Holocene thermal maximum, it was here in the central Finland, and now we have present day distribution. In Finland, we have just in the southwesternmost part, we have Corylus growing. There is some locations a little bit towards north, and at the moment the northernmost natural population of hazel is located in Leivonmäki, which is a little bit south from Jyväskylä, and these photos are from this northernmost hazel population. But using the pollen record, we can see how the distribution of some species has changed. And this is also interesting, considering that in the future, if we would have warmer temperatures, that will the range of certain species expand to the previous extents, or how much then does the human impact actually affect, and what is the role of the climate? Then some more examples. Also, we can see from pollen records if there's been some stand scale species invasion. So basically locally, how the species composition or forest composition have changed. If there's been some establishment of new species, or if there's been a big disturbance, for example, and after that there's been clear change in the first composition that has then opened and free area, so to speak, to a new species be established in the area. And here is example from my PhD thesis. I studied small forest hollows in northwestern Russia, and all of the site around 8,000, 7,500 years ago, we saw clear increase in spruce pollen, and it's kind of coincides with the start of the migration of spruce to Finland. So we got more confirmation of the actual spread of spruce trees. And in Europe, there's been, and especially in Northern Europe, Fagus sylvatica, so beech and spruce, have been studied quite thoroughly. And especially in Sweden, there has been a lot of studies of the spread and stand scale invasion of both species. And from here we can actually quite well see that spruce, here is the oldest date and youngest date, and again oldest and youngest. And this is site number 1, site number 18. We can see it from the map, 1 to 18. And from here we can see that spruce is spreading from the north, and first in the northernmost side, and then coming towards the south. But also then here again already the human impact starts to affect. But here when we then look at the beech, we can see that in the oldermost sites about 2,500 years ago, we can see the beech coming from the south and then coming to the northernmost sites only during the last 1,000 years, and some of the sites doesn't really record that well beech. So this is one example how pollen data can be used. Then a few words about how we can detect human activity from pollen records. So basically, when we go thousands of years back and there hasn't been that big human impact, we don't see that much these so-called human indicator pollens in the pollen diagrams, but more and more there is human settlement. It causes changes to the natural vegetation, changing the habitat. And then there is more and more plants and vegetation that is following human settlements. And those we call this kind of a human indicator taxa. And for example, there is some taxa like Plantago major, which you can, it spreads in this bare mineral soils, and usually you can find these taxa around some human settlement sites. So if you get this into a pollen diagram together with other indicators of landscape opening, you can think that this is the indicator of human impact in the area. Especially cultivated plants, if we get pollen from some cerealious species like rye, or if we get some other cultivated plants, Phacopyrum and so on. So we can make the deduction that probably there's been cultivation. And then usually also when pollen diagram is combined with, for example, charcoal record indicating fires, and if we get more and more fires together with cultivated pollen grains from cultivated plants, we can make this deduction that probably in the area there's been slash and burn cultivation. And also pollen analysis have been used to study archaeological sites to see what kind of plants and what kind of food there's been consumed in the past. So we can see also the human activity from the pollen records. Here are some examples how the pollen record have been used to reconstruct past human activities. In this map, there's a proportion of or sum of all non-tree species. So basically, this shows the percentage, and at this time, the greener the color, the less non-tree species, so basically more forested or most closed environment, because then when we have this yellow here, yellow color, it means that 40 to 60% of the pollen comes from non-tree species. So basically these non-aerial pollen indicating open landscape. And similar development can be seen from these maps, and these only include the pollen proportion of cerealia, so pollen from cultivated plants like rye and so on. And here, the darker the color, the bigger the percentage of this pollen type in the pollen record. So here we can see clear increase in the 15th century in the map. So during the last, let's say last 500 years, there's been clear increase in the arable land. But already much before, here we can see approximately 3,000 years ago, this a bit less than 2,000 years ago, and so on. So we can reconstruct the openness of the landscape using the pollen record. Then here again, another example from the same study that I showed already before, but showing the forest cover. But these maps are the arable land and disturbed land. So basically, when we start from the 7,000 years, we can see that there isn't that much arable land in the map. And also we can see that from the pollen diagram. And again, then when we come to towards the modern time and for the last 1,000 years, we can see increase in the map and also in the pollen diagram. And here, this pollen diagram has the land cover classes that has been synthesized from several different sites, pollen sites that you can see as a black dots here on the map. So it's kind of a spatial reconstruction also at the same time. Then one more example, just to show you this is quite recent study published last year, and just showing the origin and spread of olive cultivation in the Mediterranean basin. And here, these numbers are comparable to the pollen histograms here. And here, the dark green is areas where olive trees grow naturally, and the light green where it's cultivated, and then this hashed line shows that there's both and wildly cultivated. And if you compare the numbers of the sites and the pollen record, here we can see that in the areas where the olive trees have grown naturally, we can see that there's olive pollen here in these areas, mostly near the coast, from quite early on, from 6,000, even 8,000 years onwards. But then when we go to the sites that are in the area of only the cultivation of olive trees or further from the coastline, we can see that actually the olive pollen increases only in late Holocene, and in these sites here in central Spain, we can see that it only like less than 2,000 years or less 1,000 years, we can see that the olive pollen. But anyway, you can see that the olive cultivation has quite long history in the Mediterranean basin. So then, this is quite old study, but I wanted to show you this as we are in Helsinki, and in 1990s, there was a study done from sediment core from Töölönlahti, and from there we can quite clearly see the human impact. This shows the last 300 years in this diagram. And for example, here we can see increase in cerealia pollen. There's also pollen of other taxa that indicates grazing or otherwise human impact and increasing change and grass around the lake or Töölönlahti area. And then other big change in the 1950s happens here when they started to planting the silver willow or the willow trees around Töölönlahti, and that can be seen in the pollen record quite well. So this is also just one example how we can see the human impact in the pollen record. Then we will move on. So next, I'm going to show you one way how to actually correct the pollen proportions. So I've mentioned a few times there is some species that they are overrepresented on the pollen diagrams because they produce so much pollen, and some species that can be underrepresented. And there are different kind of models that being created to correct these errors or in the pollen diagrams. And I'm going to show you now this models that are based on the and called this landscape reconstruction algorithm. And it is a model that is developed by Shinya Shogeta. It's a Japanese scientist. And this is model that uses the pollen data and pollen persons and other pollen data to model the actual percentage cover of the vegetation on the landscape. And for these models, it includes the relevant source area of pollen. In the last lecture, I talked about the relevant source area. So basically the area that the pollen is dispersed or transported to the sampling site. Also, it takes into account a background pollen component. So how much of the pollen, how big proportion of the pollen is coming from this kind of outside of the relevant source area of the pollen to the site. Then also for every species or every taxa, there's a fall speed of pollen. So basically how long it takes that the pollen will fall to the ground. And then also it takes into account the pollen productivity estimates. So for each region or biome, for example, there is this, there's been a lot of research in these pollen productivity estimates and how those can be then used to correct the actual pollen record in the sediments. And these pollen productivity estimates, they've been done for different, or need to be done for different kind of biomes. So basically for boreal forest, there's different and for temperate forest and so on, because the pollen productivity changes depending of the area or of the habitat. Anyway, in the basic idea in this landscape reconstruction algorithm, it have two actual models. So basically they're called reveals and love. And reveals is used to make regional estimates of vegetation abundance. And for that, the pollen data comes from large sites. As you remember from the previous lecture, the larger the site, the more regional the pollen composition or the record represent. And then using the pollen productivity estimates and the relevant source area, it makes kind of reconstruction of the actual forest cover or vegetation cover. Then other steps of another model included to the landscape reconstruction algorithm is LAV, which basically is the local vegetation estimates. And for this, the data is used usually from the smaller local sites, so small peat bogs or very small lakes or forest hollows. And using these reveals and love models, they can make estimates for the actual land cover. And next I'm going to show you a few examples how it actually affects the pollen record and how it changes. So here is comparison of pollen proportions calculated from pollen record. The model estimates from reveals, so basically now the regional reconstruction, and then observed vegetation from this area. So there's two sites in Småland and then in Skåne in Sweden. And first we see here the pollen proportion. Then when we take the reveals model, so which have now the corrected values to the actual land cover, we can see that there's quite big change. For example, here we saw pine, which is quite big proportion of the whole pollen composition. And then when it's corrected in this reveals model, it's much less, whereas spruce actually is underrepresented in the actual, or in the pollen proportion, and then in the reveals model you can see the change. Also from this other side, we can see that it underestimates, it can also be seen here, but here maybe more clearly. In the pollen proportion, we have only 4% of cerealia, so cultivated plants, whereas here in the reveals modeled actual land cover, we actually have 32% of cerealia, and also the amount of grasses increases. So it indicates more open landscape. But then when we have these regional sites, and in the actual pollen record, we have a over-representation of wind pollinated tree species. But then when it's corrected, we can see that there's actually less those in the landscape. So how does then this reveals model actually compare with the observed vegetation, so the observed modern vegetation? And if we take this, we can see that the model actually suits quite well in the observed vegetation, and at least is more accurate in the presentation than the pollen proportion. So it seems that the model actually does quite good work with these corrections. Then another example, this is just, there's many diagrams from Scotland. There's two of them, this Loch Davan and Loch Maree. And here the pollen percentage are the black bars, and then the reveal-based estimates of the regional composition you can see here. So again, here the pollen proportions overestimates the tree cover, whereas then the actual model shows much higher proportions of the pollen and the open vegetation, so like heath and grasses. So we can actually see how the land cover has changed. Then just showing example that is from Sweden. This one and then the next slide, they are from the same area in southern Sweden. And this reconstruction shows now the reveal-based regional vegetation abundances here in 2 different lakes. And we can see that there's quite, let's say, even change. There's not that big variety in the sites. They are more or less similar. For example, here the pollen percentages, and then from the lakes, and then the actual reveals estimate. So basically, if we take this, for example, this pine record, here the box is Kansjön, and then the gray area is Trummen, and we can see that it kind of changes more or less here, similar, and then these are the reveals estimate. But when we compare this regional model to LAV model, which reconstruct local vegetation taken from the same area from small peat box, we can see that there's much bigger variety in the actual local forest composition and forest cover of the area. So when we have the regional estimates from reveals, we can see the actual regional land cover in the big picture. But then when we study the small lakes and use the LAV vegetation model, we can see that there's more changes in the actual vegetation in the area. So this is just examples of these land cover reconstructions. Then, well, before we go forward, there's also different vegetation models, and but now I've only chose this one, but just so that you know that this is not the only way to model the pollen and the vegetation distribution. Then I thought that I will take a few topics from the, let's say, these trends that and the research that is now done and has gained more popularity during few last years. So basically, there's been quite all the time quite rapid development in the field of pollen analysis, and as I've already said before and in the previous lecture, that all the time the pollen analysis and the analysts and paleoecologists, they are trying to bring using the paleo records and pollen towards this resolution that can be comparable to modern ecology, and also trying to get more quantitative data out and using similar statistical methods that is used in modern ecological research. And along those lines, there's been a lot of studies during, let's say, past decade concerning past pollen plant richness and diversity. Also, there's been increase in using DNA, sedimentary DNA in this past ecosystem reconstructions. Also looking into the resilience of certain ecosystems or species compared to environmental change. For example, what kind of disturbance regimes or changes in climate has probably changed ecosystems and how resilient past ecosystems have been to those changes. Then more and more there are these multi-proxy studies, so basically using different proxies, different kind of proxies like pollen, non-pollen barionomers, geochemical data, data from disturbances, and big variety of different data to make this wide range environmental change reconstructions. Also analysis with bigger data sets from this, for example, pollen databases. And when you have more bigger global data sets, you can do more wide-scale research. You can have global scale research questions in comparison if you would have just one or two sites that you would be able to analyze yourself. So these are some of the trends. Also, more and more there's collaboration of paleoecologists, modern ecologists, and policymakers. For example, you can have collaboration where there's paleo data used, for example, how different disturbances have affected different type of forests and how probably now the change in climate or environmental conditions might affect forest in different areas, and how this knowledge can be maybe used in the future to implement best, for example, forest management strategies. And at the same time, the role of paleoecology is also increased in the environmental conservation and the sustainable forest management, because when considering the conservation issues, when we can look into the past further enough, we can actually see that what has been the natural state of the habitat. And then there's these questions that what do we actually want to conserve? Is it the natural ecosystem that was at present 2,000 years ago or 3,000 years ago? Or if it's the cultural environment that was there maybe 200 or 300 years ago? So then pollen and other paleo information can be used to set this kind of baselines and get information of the past conditions. So these are the developments at the moment. And for this lecture, I've taken a few points that I'm going to present. So first of all, reconstructing the past plant richness and diversity. There's been a lot of studies into this that have been done in order to understand the past species richness and how the diversity have changed in spatial and temporal scales over the Holocene, for example. And based on the studies done so far, where the richness in pollen assemblages have been compared to modern species or plant richness, it can be said that the pollen richness in pollen assemblages more or less reflect the floristic richness, not like one-to-one, but the trend is similar. But then also there's a lot of limitations and challenges in these estimations, because as it was discussed in the previous lecture, there's many things that affect the pollen record and the pollen productivity, the dispersal, deposition, and how the pollen is preserved. So when the pollen is originating from the certain vegetation composition in the area, not all of the pollen end up in the sediments exactly in the same composition as it is in the vegetation. So there might be some overrepresented, underrepresented, some might be just lost on the process. So there's many unknowns that can be affecting. But the general trend is that there is similar changes in the past pollen richness that, or in the pollen richness that is in floristic richness, which suggests that the fossil pollen records could be used to reconstruct the past plant richness and diversity. A few examples of the studies in this area. So, for example, here is a study done by Triin Reitalu, published last year, and these maps shows the pollen richness from different pollen sites. So basically, these dots represent one site where the pollen record has been studied. And then this map presents the plant richness at the moment, or the current plant richness, the number of species. And when we compare these maps, for example, we can see that the red means most plant types of species and most pollen taxa, and then the darker blue is less species and lowest number of pollen taxa. And we can see that these sites that are here blue, they are more or less in the blue area also here in the map showing the number of species per area. And again, the pollen sites that are in red color, they are in the red area. So we can see that the trend is similar, even though obviously you can see that the number of species and taxa that is derived from pollen record is much less than what it is the actual number of species, but we can see the similar trend that the pollen richness is higher when we go to the south, and the plant richness is higher when we go to the south. And here the same study still, but just showing the correlation between the plant richness and different pollen taxa groups. So this is all taxa and then trees and shrubs. So we basically see that with trees, the plant richness and the pollen richness actually is quite well correlated. So it seems that we can use the pollen data to reconstruct also past diversity. Here is another study done from Portugal. And again, we can see there is some changes in the pollen and the actual plant. So here are the transect of sites. And again, we can see here that the trees are a little bit overrepresented, but we can still see similar kind of changes in the cover of different vegetation types in these sites. And then from these different sites and samples, they created this kind of diversity index showing the pollen or the palynological richness during the last 15,000 years. And here we can see that in the earlier Holocene, we had in this area the palynological richness, suggesting that also the species or the plant richness was higher than is at the moment. So we can also look to the long trends, how the diversity have changed using the pollen data. Then another current quite popular trend in palynological studies is using the sedimentary agent DNA that I'm going to show you a few studies. And this can be used together in pollen data to get more precise reconstructions, or there's been also a lot of studies from the sites that already previously had these pollen-based reconstructions, and then now the ancient DNA is used to make the reconstructions more precise. And when using pollen, we don't have that good taxonomical resolution, so we can get to the family and genus level, but then with the DNA we can actually get to a more diverse or more precise taxonomical resolution. So for example, if we have some plant genus or family that we only get the family level from the pollen, we can get to the species level using the DNA. And this is quite good addition. And during recent years, let's say during the last 10 years, this has been growing a lot. Here is one example published. This study is published last year. And here, this is from northern Norway. And here you can see different tree species and some shrubs. And as we see here, the gray area, it is the pollen values analyzed from the record, and then the gray orange bars are the amount of the DNA reads per sample for the species. And for here example, we can see that we have, for example, some species that there is no detection or no information in the pollen diagram. We don't see any pollen percentages from the site. But then when we have the DNA signal, we can see the change in the species in the site during the holocene. And when we look at the amounts, it kind of changes in the same lines than the DNA imprint kind of changes in the same lines as the pollen here. But this kind of shows that with the DNA, we can get more out of the samples. Then here is another, this is from northeastern Siberia, from tree-line region. And for example, here, if we look at the these blue ones are pollen, and the red ones are DNA. And this is from forest tundra, this site. And we can get more kind of red and more detailed information with the DNA. For example, with pollen, we only can go to Cyberaceae, so sedges, but then when we have the pollen, we get actually to a species level, and we can know what actual species have been on the site. Also, in pollen, we only would have this Ranunculaceae, but then with the DNA, we can see that there's actually several different species among Ranunculaceae. So we get more information out from the sediment, and actually indication of more diverse vegetation composition than what we get from the pollen. So these are now all the time developing, and there's more and more studies coming using these new approaches. But just to mention too, we don't have that much time to go through all of them. Then at the end of the lecture, I will just give you, as promised, short introduction to non-pollen palynomorphs. So basically, Non-pollen polynomorphs are this large and heterogeneous group of microfossils that are basically other than pollen. So all the time from the start of the pollen analysis, researchers have seen these other than pollen, these other microscopic objects than pollen in the pollen slides. And those have been in the interest of the researchers. And these other microscopic objects can be quite big range of different groups. It can be algae or some fungi or plants, plant parts or some animalia. So, and some bacteria or viruses. So basically it can be quite quite big range. So basically it's usually analyzed from the same samples as the pollen is analyzed. So it is kind of this complementary analysis, although lately it's been also done just solely to identify these NPPs. So a little bit of the history. It's been, as I said, around since the start of the pollen analysis. So already early 1900s, there was researchers who described these microfossil organisms from the pollen slides, but then only could say that from the 80s, it has really started growing as a study. And this is thanks to Baswan Chil, who has been kind of like a pioneer in this study, a field of study, and started systematically to record the NPPs from pollen slides. And after the 80s, there's been a substantial growth in the describing the different types of NPPs, although still many of them are unknown. And here you can see kind of the development. So it's been quite steady until the early 21st century. And let's say during this kind of statistics end to about 10 years ago. So let's say 15 years, the last 15 years, there's been a really big increase in the studies that have had this, that have included non-pollen polynomorphs as a proxy. So it's growing field of research. So how can we then use it? So basically as a proxy for past environments, so they provide a wide range of ecological information because it's a wide range of different organisms. So it also records different kind of changes in the environment and can be used to reconstruct the trophic state, change in the trophic state in the lakes, temperature or moisture conditions, dry phases, human activity, especially with reference to this non-pollen polynomorphs. There are fungi and living in dung, animal dung, so can be used to reconstruct pastoral activities. Fire can be used, reconstruct using these organisms, also erosional events. So basically they provide quite important and useful tool for reconstructing the biotic interactions. And for that, also all the time increasing in different studies. So basically then few examples that what kind of non-pollen polynomorphs there are and what can be found from the sediments. So basically when looking at the lake sediments, we can see, for example, hornwort leaf spines. So they're quite distinct looking and easy to recognize. Also, we can see some structures from water lily. And this is quite useful because both of them have quite rare pollen in the pollen record. So you might detect these parts even you don't see any pollen in the record. So then you can use this to kind of verify that this has been present at the time. And also, for example, ceratophilum, it occurs in the more warmer temperatures, and for example, can be used if you find these from more northern locations during the Holocene, for example, or some previous time periods. You can make deductions that, for example, when it's present, there has to be certain amount of the average temperatures. So it can be also used to kind of reconstruct or make indications of the possible temperatures in the area. Then also there's a lot of different algae included in the non-pollen palynomorphs, and I'm presenting here a few of them. There's different green algae that can be used to reconstruct, usually found in the lakes. So if you find these in the sediment records, you can say that there's been a lake or more weather based. There might have been some freshwater pool or so on. And then also you can use, for example, pediastrum can be used to reconstruct the nutrient stages or pH or what water chemistry. And for example, the signematotypes, they usually occur in oxygen-rich freshwater lakes. So if you find these in your sediments, you can make some indications or interpretations how the environment of the lake ecosystem have been. Then also there's cyanobacteria that can be found from the sediment samples. There's different types that you can find these fossils. And these are quite good indication of the eutrophication of the lake. And usually when compared to pollen, you can see the increase in the cyanobacteria and increase in the human indicator pollen taxa and signs of farming and cultivation. And usually these can be used to detect or interpret the increase in human impact on the area or more use of fertilizations around the lake catchment. But also then it's important to notice that eutrophication can also be a natural process. So if you go and look the Holocene records and at some point there's the eutrophication or increase in the cyanobacteria suggesting more eutrophic conditions, it isn't necessarily because of the human impact, but there might have been some natural development that have caused the eutrophication of the lake. But one way of using the non-pollen palynomorphs. Then quite commonly, the fungal spores are found in peat deposits, whereas these algae usually are in lake sediments. And when you have these fungal spores, they usually don't travel far from the origin, but they are easily breakable. And then usually when they are deposited near the origin. So it's an indication of local change and in the environmental conditions. There's many different types of these fungal spores. I'm going to show you examples of a few of them. These can be parasitic fungi or saprophytes, so basically found with host plants. Also There's fungi growing in animal dung that can be indicator of herbivores and also indicator of grazing. And also there's some fungi growing in the burned plant remains, which can be used as indication of fire. First example, I've chosen this Kretzt Maria Doista. Sorry, my pronunciation. But this is basically parasite that is growing, for example, with lime and elm. And with pollen, it can indicate the, give quite strong, for example, the decline of the taxa, or usually it kind of gives the indication that the taxa is present. Here we can see that it's usually related to the, for example, fungus. In this diagram, also there's the increase in fungus, while there is then decrease in this Ulmus antilia, as we can see also here. We can see the decrease here, increase in the other taxa. When we see the Kretz Maria Doista here, starting and then decrease here. So we can see this kind of a combination. Then fungi growing on animal dung. So one of the examples, these are also several different fungal spores, but I just now put here examples of Sporormiella and Sordaria. And usually when you find Sporormiella in the sedimentary record, it's quite good indication of higher or increased grazing, especially domesticated herbivores, usually in connection. Also, these fungi growing on animal dung, they have been used a lot in many studies to investigate the Pleistocene megafaunal extinctions when there's a decrease in these fungi, then it has been concluded to indicate also decrease of the megaherbivores. But here I've chosen one example where we can compare, for example, here is this kind of a compilation diagram. It's part of a bigger study. And here we can see this line showing the percentage of the coprophilous fungi, including Sporormiella and Sordaria and so on. And here there are some phases with this study site when there is these peaks in the fungi, and also these parts showing the grazing and the agricultural phases and grazing periods. And also this record from this one lake in this study, it's been compared to the different periods in regional regional perspective with the information from larger area around the study area, and they found that the peaks in the coprophilous fungi kind of correlates with the phases of the kind of the grazing periods in the area. So they've kind of get indication also from that side here that there's been more grazing. So one way of using these non-pollen palynomorph types. Then also these non-pollen palynomorphs can be used to reconstruct fire events because some of them are from fungi that is growing only on burned plant material, and one of the examples are Neurospora, and it's a mold that grows to a charred tree or bark after a fire event, and these are quite good indications of fire because they can only germinate after they've exposed to high temperatures, up to 65 to 70 degrees for a few minutes. So it indicates that because in natural conditions, there isn't that kind of temperatures, except when there's a fire. So can be held as a quite a reliable indication of fire. And there was conducted a research in central Finland, actually, by Norman Stevens and his colleagues, where they compared charcoal from the sediment layers with fire scars from trees, and then these neurospora fungi that they found from the sediments. And then they found quite good correlation. Here you can see the neurospora, and it correlates or occurs the same periods when there's indicator of, for example, large charcoal species showing the local fire event. But for this, we are going to talk about a little bit more in the next lectures when we are talking about more about disturbances. But just to show you that non-pollen palynomorphs can also be used for this. Then also we can use these to reconstruct erosional events in the lake catchment or near the site. And this can be done, for example, using these glomus spores, and they are this kind of a mycorrhizal fungi, and they have a variety of host species, but they can be interpreted to indicate erosion because they are in symbiotic relationship with the plant roots. So when there's erosion and when there's disturbance in the surface layer, they can start moving and then end up to, for example, in the lake within the surface runoff and so on. So here again, I've used the same study. So you can see that these are usually nowadays the case. There's multi-proxy studies using the pollen to reconstruct vegetation. Then there's different geochemical signals, and then also there's a fire indicator from the sediment, and then there's non-pollen palynomorphs. Here is the coprophilous fungi that I just showed indicator for grazing, but then there is now, they've used this glomus to indicate erosional events, and it's quite well in line with the kind of, there's more glomus proportions when there's indication of fire here and here, and also indicator of grazing. So these kind of activities that might start or get the surface moving and cause the accumulation of these spores to the lake sediments, for example. So basically here they've worked quite well in showing the erosional events. Then just showing at the end this diagram where they've used pollen taxa to reconstruct vegetation. Then there's fire dynamics, but then they've used different kind of these non-pollen palynomorphs to get more indication of the events. For example, here is the Chloetrichia. It's these indications of nutrition states. Then there's the coprophilous fungi. We can see that, for example, when we have more of this dung fungi, we have also more indication of nutritions in the lake. Also, we can see that there's increase in the grasslands and ruderal plants at the same time. And also we see here that there's increase in cerealia, so probably there's been some cultivation and so on. So then comparing these proxies, we can get much more precise and wider picture what has happened in comparison if we would just use just pollen, or if we would just use geochemical data, or just use non-pollen palynomorphs. So this is kind of nowadays quite usual that in same study and from site there's multiple proxies studied and then make this kind of a compilation of the all data. So basically still NPPs are more or less, or quite often they are complement to other proxies, but it's growing very strongly and have a big potential in the paleoecological reconstructions. And all the time there's more and more identifications and definitions of their ecology of different NPPs. So there's more and more information of different types that have been identified from the fossil samples. And there's also site that you can check if you want, when there's some pictures of non-palynomorphs and some more information. Then just quick summary of today's lecture. So basically we can use pollen records with quite a wide range to reconstruct past plant communities, changes in the population range and size. In different spatial and temporal scales, we can use the pollen record to detect human impact. Then there's different models, including this landscape reconstruction algorithm that can be used to model the actual vegetation cover. Also, the current developments are taking the pollen analysis towards more and more towards the modern ecology, that more comparable studies using different methods, combining multi-proxy data sets, using DNA records and so on. And then we can use the non-pollen palynomorphs also to reconstruct environment at their own, or then as a complementary to other proxies. So this gives quite good possibilities to reconstruct past environments. And then on online session on Monday at 10:15, I will give you the instructions for the assignment. And if you have any questions of the lecture or any other topic, you are free to ask questions then. So thank you for listening, and I see you then hopefully on Monday during the online session. Thanks.